So now we're going to go into gel electrophoresis. So gel electrophoresis, there are also several types. One of them is agarose gel electrophoresis, which uses agarose gel that is more effective for larger DNA molecules, usually after they undergo PCR. And the effective size range is between 100 base pairs and 25,000 base pairs. So as you can see, this image on the right is pretty much the agarose gel electrophoresis with all of the different bands for the different sizes of these DNA molecules. And then we also have native Page, which is polyacrylamide gel electrophoresis. So it's just a slightly different gel. And the proteins, so in native Page, it's with proteins that are analyzed in their native states. So that's when they're like fully functional proteins. And this is usually useful for comparing the molecular size divided by charge of proteins that are known to be similar in size. The reason why it's more useful for comparing these proteins when they are similar in size is because this molecular size with charge ratio can really change depending on that size. And so at least when we know that it's relatively the same size, we can, not be too worried that the charge is in a sense, or this ratio is in a sense interfering with how far these proteins are moving on that gel. And then we have Sds Page, which is, it's pretty similar. It's a similar concept, I would say, to native Page, except that the reason why there is SDS is because SDS is a detergent that denatures the proteins and coats all the proteins in a negative charge. So they only are separated by molecular mass, but also at that earlier protein structure stage, because when SDS denatures the proteins, they get rid of all of the non-covalent bond interactions. And so then you're left, and they're all also coated with a negative charge. So you know that their charge is the same, and everything that they would be separated on is not quite dependent on charge anymore, but only by the molecular mass. And this is different from native Page because we can't see the functional protein itself from the gel because we are denaturing the protein. And this is kind of an image of Sds Page on the right. I feel they kind of look similar, but that's pretty much, I guess, how a lot of gels look. And then isoelectric focusing is when proteins instead are separated by their isoelectric point. So if you guys remember from the chemistry lectures, or possibly physics, so chem phys lectures, the isoelectric point is that pH where these proteins have a neutral charge. And so based on all of these charges, they have usually positively charged proteins moving towards the cathode of the gel, and then negatively charged proteins moving towards the anode. And the protein migrates toward until it reaches a gel region where pH equals pi, which is the isoelectric point of the protein. And so you can see in this image to the right, it's kind of, you can see how like the pH is a little bit higher at 6 at the top with that positive charge at the end, and then pH equals 2 here at that negative charge at the bottom. So these proteins are being separated based on that. So now we are coming up to the first poll question. We have 100% saying D. Okay, so the correct answer is actually B, cysteine, and I'll go into that now. So, yeah, so the question asks, a bond formed between which of the following amino acids is disrupted in reducing Sds Page gel electrophoresis? And so this question I was trying to sorry, this question involves a little bit more, I guess, extrapolation of the knowledge that we know about Sds Page. So as we remember, Sds Page has that detergent Sds, which denatures proteins, so that any non-covalent interactions would no longer be interacting in the protein. And so usually the proteins end up back in, I would say, the primary structure. And so when we're looking at all of these amino acids here, one stands out pretty well because we know that within the tertiary protein structure, a defining feature of it are the disulfide bonds, which are created by the two cysteine amino acids bonding between each other, right? And so that's why B is the correct answer, because most likely when we're reducing the proteins with SDS, it will most likely not have that tertiary protein structure feature. And so that's why the answer is B.